Hello everyone, welcome to Get My Class. Let's solve this question which says source of complementary RNA for RNAi could be option A from an infection by viruses having RNA genome. Option B mobile genetic material that is transposons that replicate via an RNA intermediate. Option C from complete genome of the host cells and option D both A and B. Now, the question says source of complementary RNA for RNAi could be. So, first of all, what is RNAi? RNAi. RNAi is nothing but interference RNA. Okay, interference RNA. So, why are we calling it as interference RNA? Here, it is actually a defense mechanism. Mechanism which occurs in all eukaryotes. So, this interference RNA is produced in all eukaryotes as a defense mechanism. Okay, basically, it is to prevent the expression of unwanted gene. Expression of unwanted gene. Unwanted gene. So when we say gene, okay, of course we know that genes are the parts of the DNA, right? They are present in the DNA. So this is the DNA. You have specific sequence which codes for a protein, and that is why we call it as a gene because it will code for a particular protein. So of course you have the genes which are active, which can code for protein. There are other genes which may not be active, and those can be considered as junk genes. But here I'm talking about the genes which are active, which can express. So when we say express, suppose this is the gene. Okay. So of course the first step for expression is the transcription, right? That is the formation of mRNA. And the second step is protein formation. Okay. So if there is any unwanted protein, this may cause disease or any syndrome. To avoid the expression of unwanted protein. So this unwanted protein can be of a virus because of viral infection. If this unwanted protein can be of a transposon, which is not necessary, whose expression is not necessary. So this protein, if it is not needed in our body cells, there is a mechanism which produces the complementary RNA to this mRNA. Okay, that is this mechanism. So what will happen? This becomes double-stranded RNA, and double-stranded RNA cannot be recognized by ribosomes and it cannot be translated. This is what is called as RNA silencing. How is it happening? By converting single-stranded mRNA into double-stranded mRNA. That is what is RNA silencing. Okay, and this mechanism is called as RNAi. Okay. Now, if we have to silence a particular RNA or particular expression of a particular gene of a viral infection or a virus or for a transposon, we have to use the same as the source of RNA. Okay, that means if we are talking about the silencing of a viral gene, we have to look for the RNA of the viral gene. Then we can use it as the complementary strand, and we can convert the actual viral mRNA into double strand, and that is what will cause RNA silencing, and there will not be expression of protein. Now let us see. The question says source of complementary RNA. So if you have to suppress the expression of a particular RNA, what will you consider as a source? Because of course that source will be complementary only, right? Complementary to that RNA, because double strand can be formed only when you had complementary RNA. Okay, so let us see. Option A says from an infection by viruses having RNA genome. Yes, this is absolutely right. If you have to suppress the expression of the viral reproduction, you need the RNA of the same genome, right? Like RNA of the same virus. Only then you can make it double-stranded and the expression will be stopped. Hence, this is correct. Option B says mobile genetic elements transposon that replicate via an RNA intermediate. Definitely, the this transposons also can be suppressed. Like if we don't want the expression of transposons, and in nature also this is what happens. Transposons, if they are not important, if they don't express, because there is RNA silencing. To silence such transposons, the RNA intermediate can be considered as the complementary RNA itself. Okay. Hence, option A and B can be utilized as a source of complementary RNA for RNAi. Option C says from complete genome of the host, not the complete genome of the host, because complete genome, then the entire genome itself will produce, you get many number of RNA molecules from complete genome, because each individual gene will code for mRNA. Individual DNA will code for individual mRNA. Okay, that means I'm talking about individual gene of the DNA will code for individual mRNA. So always remember one gene, when it is transcribing, that will code for one mRNA. So in a genome, you have many number of genes. So how many whatever numbers of genes are there, so many numbers of mRNA will be formed. Hence, this cannot be used as a source for RNA, for a particular RNA. And option D says both A and B, that is absolutely right. So let us conclude. But the answer is option B, that is both A and B can be used as source of complementary RNA for RNA interference. I hope you understood the explanation. Thank you.